Thank you very much, Catherine, for this nice introduction. And before I start, I would like to thank the Plant Section Committee for choosing me for this award. This is really a great honor. Thank you very much. And I've seen the former medalists, and I just hope I can keep stepping into their footsteps. They're really fantastic scientists. So, what is Arbuscular mycorrhiza? It's the most important symbiosis on Earth. And I know almost most of us would say that about their own research subject, but for mycorrhiza it's really true, because it occurs with about 80% of land plant families and with fungi of the Glomeromycotina. So 80% of land plant families is a lot. Yeah? It provides important ecosystem support. For example, we think that tropical rainforests could not exist without this symbiosis, as they grow on very shallow soils in which nutrients cycle rapidly, and the fungus is a great help for plants to take up these nutrients before they leach out of the soil. It is approximately 400 to 450 million years old, and the oldest land plant fossils already contain the fungus in their roots or in their stem-like structures, and therefore we think that Arbuscular mycorrhiza fungi, together with other fungi that are now coming up in the literature belonging to the Mycorrhizae, help plants to make the step from water to land. Because at that time, plants didn't have complex root systems to take up nutrients. And this already brings me to the advantage of this symbiosis for plants, which may be the reason why it has been conserved for so many hundreds of millions of years. So the fungus develops this highly extended and branched hyphal network in the soil, which reaches beyond the nutrient depletion zone, which forms around the root due to nutrient uptake by the root. And these hyphae take up mineral nutrients, transport them into the root, and then release them at these highly branched fungal structures, the arbuscles, which have a very extended surface area. And you can really see the difference when crop plants grow on low nutrient soils, like this soil here in Australia. They grow much better in presence of the symbiosis than in the absence. But not only the plant profits, also the fungus gets something. It gets carbon, which has been fixed in photosynthesis. And the textbooks say that these are only hexoses, sugars. But I will tell you today that the fungus also receives lipids from the plant. So for the symbiosis to occur, the root needs to be colonized. And this is an interesting stepwise developmental process which is largely under plant control and is one of the main research interests in my lab. So this is a longitudinal section through a root with the rhizodermis and the cortex. And here is a fungal spore which germinates in the rhizosphere and enters into a bidirectional molecular dialogue with the plant. Plants grown at low phosphate exude strigolactones. This is a plant hormone, and strigolactones induce vigorous growth and hyphal branching of the fungus, and we think that this enhances the chance of encounter with the root. In turn, the fungus releases chito-oligosaccharides and lipochito-oligosaccharides, and these also induce symbiotic responses at the plant site. Now, when the tip-growing hypha reaches the surface of the root, it differentiates into a hyphopodium, and this now leads to very interesting subcellular rearrangements of the underlying cell, leading to a cytoplasmic bridge across the vacuole, which is lined by membrane, cytoskeleton, and ER, and guides the fungus through the cell. And when the fungus then reaches the inner cortex, it exits the symplast and spreads apoplastically, so between the cells, and inserts these highly branched arbuscles inside cortex cells. And in my lab, we care about different aspects of this developmental process. So we work on how is quantity reached, and quantity is a function of initiation and spread. And to understand quantity, we mainly work with the function of the karragine receptor complex in regulating colonization. We also work with arbuscle formation. So what are the plant molecular players that regulate and execute arbuscle formation? Not only because arbuscles are basically the key structure of the symbiosis because they release nutrients to the plant cell, but also because this is a very interesting cell developmental process on the plant side. and it is actually transient. So after a few days of maturity, the arbuscles collapse again and disappear from the cell, and the cell goes back to its original state. So it's an interesting model of transient cell development. We also care about application of the symbiosis, and here we want to understand the genetic underpinnings of functional compatibility. And this is a phenomenon that is observed and relates to the fact that it depends on the plant-fungal genotype combination how much the plant profits from the fungus. And if we could understand the genetic basis of this, we could on one hand predict which plant-fungal combinations would work best in the field, and on the other hand we could breed for mycorrhiza optimized plants for better sustainable agricultural practices with reduced chemical fertilizer input. But today I would like to talk to you about arbuscle formation and the most symbiotic topic in my lab, and this is transfer of lipids from the plant to the fungus, because the symbiosis is based on nutrient exchange. Arbuscle development is also a stepwise process which can be dissected by plant mutants, and I don't want to go into too much detail here. I just want to tell you that first the cell rearranges again, then the fungus inserts a trunk, produces first-order branches, and through higher-order branching it finally fills the whole cell, and then it collapses. And I also would like you to remember that the arbuscle is always surrounded by a plant-derived so-called periarbuscular membrane, and this membrane hosts a specific protein composition which so far has been found only in this membrane. And interestingly, the localization of these periarbuscular membrane resident proteins depends on the promoter of the gene which encodes them, and this has been found in the lab of Maria Harrison in the US. So please remember this. If you are expressed early as a gene during arbuscle development, the encoded membrane protein will localize to the peripheral membrane, and if you are expressed late, the encoded protein will localize to the periarbuscular membrane, and this will become important later in my talk. Now, for arbuscular mycorrhiza development, we mainly work with Lotus japonicus. This is a model legume and has two advantages. It also forms root nodules, and we can compare the two symbioses, arbuscular mycorrhiza and nodulation, because they have a lot in common. And we can also perform hairy root transformation, which generates chimeric plants, so a non-transgenic shoot and transgenic roots, which you can see here in fluorescent red. And this method is very rapid. Within 4 weeks, we can do experiments already as compared to 1 year for stable transformation. And we have performed a forward genetic screen and found Lotus mutants with a defect in arbuscle development. So here the fungus is stained with a lectin called wheat germ agglutinin that binds to the fungal cell wall, and this is linked with an Alexa fluor to visualize the fungus in the confocal microscope. Wild-type arbuscles fill the complete cell and are highly branched, whereas in the mutants, the arbuscle development is attenuated and the arbuscles are small and stunted. Now, we identified the mutations by a combination of mapping and next-generation sequencing, and we could also restore the wild-type arbuscle phenotype by reintroducing the wild-type copy of the corresponding genes. Now, one mutant is called disorganized arbuscles, or DIS, and the mutation of the other mutant had been already identified in Medicago before, or the gene, and that mutant had been called reduced arbuscular mycorrhiza 2, so we called this mutant RAM2 as well. And both genes encode for lipid biosynthesis enzymes. This encodes for a beta-keto-acyl ACP synthase 1, or in short Cas1, and RAM2 for a glycerol-3 phosphate acyl transferase 6, or GPAT6. Now, Cas1 is involved in fatty acid chain elongation, or performs fatty acid chain elongation in the plastid from C4 to C16, leading to palmitic acid. And RAM2, or GPAT6, is predicted to localize to the ER and to link fatty acyl-CoA to glycerol to produce beta-monoacyl glycerol. Now, why would we need lipid biosynthesis genes for mycorrhiza? And before coming to that, I would like to show you another phenotype that we found. So the mutants do not only not support arbuscle branching, but also the quantity of colonization is reduced. Here you see percent root length colonization of the wild type and 2 allelic dis mutants, and they can still form quite some internal hyphae, but the amount of arbuscles is strongly reduced, and the vesicles are almost down to 0. I didn't tell you yet what vesicles are. These are these balloon-like structures some fungi form, and they are filled with lipids. So it looked a bit like the fungus is deprived of lipids in the mutants. And from now on, I will tell you only about this because we have exactly the same data for both mutants or both genes, and I don't want to overload the slides. So it was known before that RAM2 is a mycorrhiza-specific copy of a housekeeping GPET6. So for example, in Arabidopsis, GPET6 is involved in cutin monomer biosynthesis in the flower. And in mycorrhiza-competent plants, there's an additional copy which is responsible for mycorrhiza. So we wanted to know the phylogenetic pattern of this as well. And we made a phylogenetic tree of the Cas1 proteins in land plants. And you see here that this tree falls into 2 clades. One we call the Cas1 clade because it contains the product of the single copy Cas1 gene in Arabidopsis. Therefore, we suspect that the members of this clade have housekeeping functions, and there's also a lotus copy. And then we have the dis clade, and in the dis clade we only have representatives of mycorrhiza-competent dicots. And interestingly, the monocots cluster all in the Cas1 clade. So this is in yellow, the monocots, or better to say, the grasses. And in both clades, we have a representative of the gymnosperms, indicating that the monocots may have secondarily lost this, maybe because their housekeeping Cas1 acquired a new regulatory element in the promoter to make them also functioning in mycorrhiza. Still, when we found this is quite some time ago already, we thought maybe this is not a canonical Cas1, but maybe it acquired a new function. And we tested this by cross-species complementation with Arabidopsis. So there's an Arabidopsis knockdown mutant. There's no knockout mutant because it would be lethal. And this suffers from perturbed chloroplast division and a small rosette. Now, when we introduce the wild-type dis gene into this mutant, we get big wild-type-like rosettes back, and our negative control was the mutant dis gene, which does not restore rosette growth. We also did the opposite and introduced the Arabidopsis Cas1 into lotus roots and could restore full arbuscle branching, indicating that this functions as a canonical Cas1. Now, why do we then need a special gene only for arbuscular mycorrhiza? Probably for the expression pattern. So we wanted to know how does the promoter activity pattern of this look like in mycorrhizal roots. And this has always been very frustrating for us because we cannot yet transform the fungus, so we cannot make a fluorescent fungus and look at a co-localization of proteins or promoter activities with fungal structures in living roots. So either we stain the root and kill it and then we see the fungus, or we have a live root and we can't see the fungus. So we wanted to find a way around that. And now remember the protein localization that depends on promoter activity. So we wanted to find a promoter which would drive gene expression such that we can send a fluorescent protein to the apoplastic space which surrounds fungal structures if we tag it with a signal peptide. And luckily we found such a promoter. This is the promoter of SBTM1, a mycorrhiza-induced subtilase. And when we fuse it to a signal peptide M-cherry, we can beautifully visualize the silhouette of the arbuscle. So the M-cherry is now sitting in the periarbuscular space, the apoplastic space between the arbuscle and the plant cell. And as you can see here, promoter activity shown here is nuclear localized YFP, perfectly correlates with arbuscle containing cells. And we could even dissect the different stages of arbuscle development. So we could see promoter activity at the pre-penetration apparatus stage, growing arbuscles, fully grown arbuscles, but not anymore in cells containing collapsed arbuscles, indicating that this gene is really expressed during arbuscle development, which fits with the phenotype. We could also show that arbuscle containing cell-specific expression of this is sufficient to restore full arbuscle branching and also full amount of colonization by complementing the mutant with a promoter that is specifically expressed in arbuscle-containing cells. And this shows us that intact arbuscles are really important also to support full colonization of the root, and there's a dependence for the fungus on arbuscles. Now, I told you that this encodes a Cas1 enzyme. Its function is specific to arbuscle-containing cells, and it is required for vesicle formation, so the formation of these balloon-like structures which are full of lipids. Now the question was, what are these Cas1-derived fatty acids good for? Of course, they may be used for synthesis of the periarbuscular membrane, because all of a sudden the cell needs a lot of membrane material. But they could also be a fatty acid-derived signal molecule, or they could be used for fungal nutrition. And this was, of course, our favorite hypothesis, because it would, if it's true, overthrow textbook knowledge, and we always like to do that. And there was also were some hints in the literature. So the same people who had actually found that only sugars are transferred from plant to fungi found that de novo fatty acid biosynthesis inside the fungus only occurs when the fungus is inside the root and never in the extraradical mycelium. So this was already a strong hint that these fatty acids may not even come from the fungus. And then while we were working on this project, our collaborator Peter Derman from Bonn University looked carefully at the first sequenced arbuscular mycorrhizal fungal genome and found that it is lacking genes which encode cytosolic fatty acid synthase subunits. And the cytosolic fatty acid synthase is usually the responsible for bulk fatty acid biosynthesis in fungi. So this already was a very strong hint that the fungus may be dependent on fatty acid supply by the plant, because these fungi actually store a lot of lipids in their vesicles, but also in their spores. And they mainly store it in the form of triacylglycerol decorated with palmitic acid, and this is the product of this. And they also introduce a fungus-specific desaturation at the omega-5 position, leading to 16,1 omega-5 fatty acids. And this comes handy because it can be used as a fungal-specific fatty acid tracer. So we wanted to understand whether indeed the plant transfers fatty acids to the fungus. And we tested this directly using stable isotope labeling. And this is not so easy, because you need to imagine the root and the fungal hyphae are all in the soil, and how do you separate it all? So we decided to use an in vitro system in split petri dishes that you can see here. But the problem is now that Lotus japonicus doesn't colonize very readily on a petri dish. So we needed a nurse plant, and here we used a carrot hairy root, which is often used to propagate the fungus in vitro, also by companies. So we used this hairy root, inoculated with the fungus, waited until the whole plate was colonized so the fungus can jump over the wall. And then we added 2 lotus seedlings and labeled them with 13C labeled glucose. And then we traced the heavy isotope in the 2 marker fatty acids, 16,0 and 16,1 omega-5. And here I show you just the amount of label in 16,0 omega-5, or the percentage of label. Here are control roots without fungus. So this is now inside the root of carrot. We also labeled carrot. I will tell you later why. Lotus wild type, dis and ram2. Here are mycorrhizal roots. Nothing exciting is happening. But if we now look at the extra radical hyphae, we see big differences. So here we really extracted the hyphae separately from the agar. This is the fungus growing in association with carrot root, a lot of label, with lotus wild type, also some label. And when it grows in association with the mutants, there's hardly any label to be found in the extraradical hyphae of the fungus, similar to when the fungus is labeled in absence of the plant. But still, this doesn't tell us if the fungus received fatty acids from the plant, or if it received sugars and then made the fatty acids by itself. So we turned to isotopologue profiling. And isotopologues are versions of the same molecule, but with a different isotope composition. So for example, for palmitic acid, you have here one with only 12 C atoms, then there could be some with 1 13C, 2 13C, 3, and so forth, until all C atoms are heavy, are 13C isotopes. And you can then quantify these different isotopologues by mass spectrometry, and this results in a characteristic pattern, and this pattern can be used to trace metabolites through pathways or between organisms, as we did now. So here you see 3 independent biological replicates in the isotopologue pattern of 16,0 fatty acid. On the left is the colonized root, and on the right the fungal mycelium, which is connected to exactly that root, so the extra radical mycelium. And you see that the isotopologue pattern is exactly mirrored, the isotope pattern of the plant is exactly mirrored in the fungus. And this is also true for the fungal specific fatty acid. This is now the fungus inside the root and the fungus outside the root. It exactly recapitulates the plant pattern, indicating that the plant made the fatty acids and provided them to the fungus. However, if you are critical, you can still say this is not enough, because there may be a biophysical law which always leads to this pattern, independent of whether the plant or the fungus made the fatty acids. So we need to find another system which would result in a different pattern, so that we could see again if the pattern is conserved in the fungus. And for this we turn to the carrot root, because we figured that the carrot root doesn't have a shoot, so sugar uptake doesn't compete with photosynthesis, and we should therefore get more uptake and a higher order labeling pattern. And indeed this worked. So here you see the labeling pattern of the carrot root and the connected extra radical mycelium. And again, the pattern in the fungus is completely conserved, and we even have some variability between the different replicates, and each time, independent of how the plant pattern looks like, the fungus has the same pattern. And this was now really strong evidence that the plant provides fatty acid to the fungus. And we also looked at the mutants. You see them here. And the 16,0 fatty acid pattern in the root very well mirrors the pattern in the wild type. And this is also important, because you could criticize that the pattern inside the root is dominated by the fungus, because the fungus has these lipid-filled vesicles, and then again we couldn't conclude anything. But it seems really the root pattern, because the mutants mirror it, and then the pattern breaks down in the fungal hyphae when they are connected with the mutants, and you see that most drastically even for the fungal-specific 16,1 omega-5 fatty acids. Here already inside the root, the fungus cannot maintain the pattern that the plant provides. So we conclude that in colonized cells, so this is a cell with an arbuscle, there is a normal lipid biosynthesis pathway here shown in black, but there is a superimposed mycorrhiza specific lipid biosynthesis pathway which can be induced on demand. And this is, we know only 3 components of that so far. DIS and RAM2 I showed you, and some other laboratories have found a thioesterase which terminates fatty acid chain elongation, and it is called FAT-M. Now the question is, which lipids are transferred? So we only looked at the fatty acids as markers, and how are they transferred? And we don't know yet, but we suspect that beta-monoacylglycerol is a good candidate as a vehicle for fatty acids into the fungus, because not only because it's the product of RAM2, but also because this is not so much used for the plant's own lipid metabolism. So it would be a clever way to withdraw fatty acids from the plant's metabolism. Now, how do the lipids get out of the plant cell? Maria Harrison has identified 2 half-ABCG transporters called STAR and STAR2, which localize to the periarbuscular membrane, and when they are knocked out, they lead to similar phenotypes as the lipid biosynthesis genes. And they belong to a family in which there are other transporters which transport lipids. For example, the human cholesterol transporter is an ABCG transporter. And then we have to find out how the lipids get across the periarbuscular space and into the fungus, and there are no hints so far. Now, while we were working on this, there were also other laboratories working on that problem, and we learned that only quite late. So Maria Harrison provided beautiful circumstantial evidence through very detailed phenotyping of lipid biosynthesis mutants, mycorrhiza-specific lipid biosynthesis mutants, that there must be lipid transfer. And there were 2 papers in Science by Ertor Wang from Shanghai and by Giles Aldred and Peter Eastmond from Rothamsted and Norwich, who also showed that fatty acids are transferred from the plant to the fungus. And they used a different method than us. They used a synthetic method. So there is a FATB, a thioesterase from Umbellularia californica, which terminates fatty acid chain elongation already after 12 C atoms to form lauric acid. And this lauric acid does not occur in Medicago and neither in the fungus. So they expressed this gene in Medicago and could then trace lauric acid and found it also in the fungus, which is also very strong evidence that fatty acids can be provided to the symbiont. And I'm really happy because we have now 2 different methods showing the same thing, and I think it's really true. Now, why would the fungus be so stupid to lose its fatty acid biosynthesis genes and become entirely dependent on the plant? I would say because it could, because probably or most likely fatty acid transfer from the plant to the fungus evolved first, and then the fungus could lose the genes as a consequence. Now the question is, why does the plant provide fatty acids to the fungus and invest so much energy? And I think because it's also an advantage for the plant, because sugar transfer plus fatty acid biosynthesis is a huge energy expenditure for the fungus, and receiving the lipids directly is much easier and would fuel much faster growth of the fungal hyphae. And this then also feeds back on the plant because more hyphae can take up more mineral nutrients and provide them to the plant. And with this, I'm at the end, and I would like to thank my fantastic lab, especially Andreas Keimer, who really drove this project, and Priya Pimprika, who cloned RAM2 and who performed the beautiful confocal microscopy, co-localizing arbuscles with promoter activity. And I thank our collaborators, Pierre-Marc Delot, who made the tree, Peter Dörmann's lab, who performed lipidomics for us, which I didn't show today, Wolfgang Eisenreich performed the isotopologue profiling for us, and Martin Paniske and Trevor Wang provided materials. Thank you for your attention.